Markets Lab in the Food Science and Technology Department here at UC Davis. I work under the guidance of PhD student Jung-Yao Xue, and in our study we compared 3 quantification methods used in bacterial profiling. So the first objective of our study was actually to compare 3 quantification methods: quantitative PCR, the ion-torn personal genome machine, and plate counting to assess if current methods produce consistent results with one another. The second objective was also to observe if there was PCR bias or bias introduced by the different DNA extraction kits that we used. So to accomplish these goals, we made a mock community of 9 different strains of bacteria. So this mock community consisted of 9 different strains of bacteria, all from dairy samples. So their names are actually shown in the table over here. They all play different roles in the dairy sample, including like maintenance of gut health, or like food technology or spoilage. So for more than 150 years, researchers were confined to biochemical or colony-based methods such as gram staining or plate counting in order to grow and identify their bacteria. However, with the recent biotechnology explosion, methods such as PCR or gel electrophoresis or high or low throughput sequencing methods have made more of an impact on bacterial studies. The problem with this, however, is that we have to verify the assumptions made during the development of this technology and also to assess the accuracy of the technology. Previous studies, such as the Jumpstart Consortium Human Microbiome Project, have actually used a mock community to compare different quantification methods. Previous publications have also compared DNA extraction kits and different quantification methods and shown that they produce inconsistent results. So the 3 methods that we used were ion-torn, qPCR, and culture-based enumeration method. So this method, or plate counting, actually has many disadvantages, including less than 1% of all bacteria is actually culturable. It can only detect live bacteria. It takes an extensive amount of time and effort to do this method, and it cannot profile a microbiome. One advantage it has, however, is that it is a simpler protocol compared to other methods that were recently developed. So the way we built the bacterial profiles using this method was we performed CFU enumeration, which is just a fancier name for plate counting. We used serial dilution on the culture broths, and then we multiplied by the CFUs by the number of 16S rRNA copies for each strain. And we were able to find the number of 16S rRNA copies for each strain by searching databases such as the NCBI databases. The quantitative PCR is a molecular method that was recently invented in the 80s. So it has many more advantages compared to plate counting, including it's a real-time quantification method, so we can actually quantify the DNA amount as it amplifies. It's orders of magnitude more sensitive than plate counting. It can detect both live and dead bacteria, and it can determine bacterial profiles. Some disadvantages it has, though, is it cannot sequence a microbiome, and it can't identify bacteria without the appropriate primers. It's also a low throughput method. So the way we built the profiles for qPCR is qPCR can detect the fluorescence intensity of samples. So we loaded the DNA samples with some fluorescence dye. It was called cyber green-based fluorescent dye. And then since fluorescent intensity is correlated with the cycle threshold, and the cycle threshold is correlated with the DNA concentration, and the DNA concentration was correlated with the number of 16S rRNA gene copies, we were able to use the qPCR data to find the number of 16S rRNA gene copies for each sample. And we built the bacterial profiles from this data. So we target the 16S rRNA gene because it contains both conserved and variable regions. So this actually helps with the sequencing for ion-torn sequencing and the qPCR. It's also present in all bacteria because it's essential for protein synthesis. So the high-throughput sequencing methods, such as the ion-torn, are actually the most recent methods. Some advantages are they're extremely efficient, and they can sequence and profile microbiomes and identify bacteria in a massively parallel way. Some of the disadvantages are, though, it's unable to acquire high-resolution taxonomy. So we built a bacterial profiling using these methods by, because ion-torn can actually sequence the DNA, we targeted the 16S rRNA gene. So the ion-torn can sequence the 16S rRNA gene, and then we can build the bacterial profile automatically with the machine. So previous work done in the lab was bacterial cell enumeration, or plate counting, DNA extraction using either the mobile power food microbial DNA isolation kit or the Magmax total isolation nucleic acid isolation kit. The PCR controls were created, and also the ion-torn sequencing was run. So 2 treatment groups, one for each DNA extraction kit, were created so they can be compared using the 3 quantification methods. So we actually ran the PCR controls, the mobile extracted DNA and the MagMax extracted DNA through the qPCR, and this gave us the cycle thresholds for each of the values. We also ran pure DNA extracted using the mobile kit, and we knew the concentration of this DNA, so we were able to create standard curves because it gave us the CT values, and we were able to calculate for the 16S rRNA gene copy number. So we built one of these, the standard curves, and we used these to find the number of 16S rRNA copies for the unknown samples using the CT values. So we did this for all 9 strains of bacteria. We repeated the same protocol 9 times, and we were able to create, we were able to find the absolute number of 16S rRNA copies, which is shown in the figure on the left. So the y-axis is the number of 16S rRNA copies, and the x are the different bacterial strains. So we added up all of these numbers and then divided one of the bacterial numbers of 16S rRNA copy genes by the total sum to determine the proportions, which is shown in the figure on the right. So for our results, we ran a Kruskal-Wallis test based on the pairs of different quantification methods, such as like qPCR versus ion torrent, or the ion torrent versus plate counting, or qPCR versus plate counting. And we found that there are significant differences between the ion torrent versus the qPCR, or the ion torrent versus the plate counting. However, there are no significant differences between the qPCR and the plate counting, which indicated that the ion torrent method needed further optimization in the future because its results did not match the results of the older established methods. Additionally, these Kruskal-Wallis tests these Kruskal-Wallis tests also told us that the methods in themselves were inconsistent because there are many exceptions to these results. So this is actually the figure that we built. So each of these quantification methods has the proportions built, like I just showed you, from the qPCR, the ion torrent, and the plate counting. And we built it this way so we can actually compare the different proportions between the 3 different methods of quantification. So for the PCR controls, we also ran Kruskal-Wallis tests, and we found that there are no significant differences between the qPCR and the plate counting and the ion torrent and the plate counting. So there's basically no PCR bias in our study. However, it was interesting to note that there were significant differences between the qPCR and the ion torrent, but that isn't as important as the results for the qPCR versus the plate counting and the ion torrent versus the plate counting. We also used Kruskal-Wallis tests to compare between the mobile extracted DNA and the MagMax extracted DNA, and found that there are no significant differences between the results. So in conclusion, there are inconsistent and differing results for all 3 methods, but especially for ion torrent sequencing, which indicates that it needs further optimization. There is no PCR bias observed in this study, and there are no significant differences between the MagMax and mobile DNA extraction kits, indicating that the choice of the DNA extraction kit did not matter as much. So for future applications, since this study contributed toward a standard and reproducible protocols in bacterial profiling, so this study contributed toward a reproducible protocols and also a standard in bacterial profiling. So future research should focus on creating a standard and also optimizing the high throughput sequencing methods, which would result in a number of applications in the future. Examples of which would be like for pharmacies targeting infections better by distinguishing the consortium of the beneficial bacteria from the harmful ones, and also being able to assess the quality and safety of fermented foods and dairy samples before they're consumed or before they spoil. So I would like to acknowledge Jung-Hyun Xue, my mentor, Dr. Maria Marco, my principal investigator, Annabelle Yu of the Marco Lab for her help and support, Stephen Ramsey, my counselor, Dr. Rick Pomeroy and Megan McKenzie, the YSP directors, and the rest of the Marco Lab and YSP scholars. Also Brian, because he made me. This is a list of my references. Thank you for listening. Are there any questions? Alex?
Why is plate counting considered the gold standard?
Oh, well, there are many papers about it because it's like the oldest method, right? So it can only be used for like counting bacteria. So in our case, we used it to actually help profile a mock community because we actually had databases that had the number of 16S rRNA copies. But it's the gold standard because it's the oldest method, basically. Alright.
How do the control and variable regions of the 16S rRNA assist with the qPCR ion torrent?
Okay, so ion torrent, actually, we perform conventional PCR on the DNA by targeting the conserved regions because we need to amplify the 16S rRNA genes. And then the ion torrent can actually distinguish between the bacteria and actually build the profiles. But for qPCR, we have to do the strains separately. So then we have to target the the variable regions which differ between each strain. So by targeting those with special primers for each bacteria, we're able to do the qPCR. David?
What does it mean by low throughput and high throughput?
So high throughput is like it can quantify like the data like many samples like at the same time. Like while low throughput can only do like one sample at a time. Nick?
So for the 3 different methods, they all had like somewhat differences in like what they produce as results.
Mm-hmm.
So what is like the most accurate, most ideal for research purposes?
Well, so the thing is the theoretical results that we use were the plate counting because they are the gold standard. So we compared all 3 methods. Like we're not sure which are like completely correct because everything has like has a bias, right? But then we found that the qPCR and the plate counting had no significant differences, while the ion torrent did. So we're assuming that the ion torrent needs like more optimization in the future because it's also a newer method. So I would say like the qPCR and the plate count. Okay.
But is it possible that the qPCR like counting can be like incorrect while the ion torrent can be correct?
That is possible, but it's more likely like the other 2 methods because the ion torrent is like new technology. But who knows? It could be like more correct. Any other questions? Thank you for listening. Introduce my colleague Nathan Lee, who also works in the Market Lab.
Nathan, we're running about 3 minutes ahead of time. Are your parents watching on video, or is everybody who's supposed to be here here?
Yes.
Good afternoon, everyone. My name is Nathan Lee, and today I'll be discussing the stress tolerance of Lactobacillus plantarum, a certain species of lactic acid bacteria. I'm proud to work under the tutelages of Dr. Maria Marco and Annabelle Yu in the Food Science and Technology Department here at UC Davis in the Marco Lab. Now, let's begin. So what are lactic acid bacteria specifically? Lactic acid bacteria is a low guanine cytosine content, predominantly non-motile bacteria that and is gram-positive. However, more important to my project as well as to society, lactic acid bacteria is responsible for food fermentations. In our project specifically, we researched Lactobacillus plantarum, a certain species of lactic acid bacteria. So why exactly do we study Lactobacillus plantarum? Well, we studied it for a couple of reasons. One, it's Lactobacillus plantarum is found in fermented foods and the animal digestive tracts. L. plantarum is a very ubiquitous bacteria and is found in multiple environments. Number 2, L. plantarum has already been significantly researched. Therefore, we and my team were able to understand the genetic background going into our project. And finally, our lab, my lab mentor Annabelle and her previous graduate student already studied L. plantarum with the osmotic stress and the heat stress, and so it's made sense to stick and work with plantarum. Now, the specific aim of our project was to look and analyze the different environmental stresses in which Lactobacillus plantarum could survive. And so we looked at three different environments. One, an acidic stressful environment. Number two, an ethanol concentrated environment. And number three, an oxidative stressful environment. Now, how we tested the acidic tolerance of L. plantarum was we initially grew overnight cultures of Lactobacillus plantarum isolates, as you can see here in this table, and then we washed these bacteria twice in phosphate buffering saline, or PBS in the scientific community. And after that, we grew the plantarum isolates in pHs of 3.5 to simulate the acidic environment, as well as a control of a pH of 6.5. And we examined the growth of the bacteria over a period of 48 hours using optical density at 600 nanometers. Now, as for ethanol tolerance, we practically did the same thing. So yes, like acidic tolerance, we grew the overnight strains, and then after that we also washed in PBS again. However, the only difference is that this time we grew the bacteria in 8% ethanol concentration, and as a control, we grew it in water, or in effect 0% ethanol concentration. And so we also examined the growth of the bacteria over a period of 48 hours, also using optical density at 600 nanometers. As for the oxidative stress environment, we again grew overnight cultures of the plantarum strains, and then we also washed it again in PBS. However, this time we immersed about 10 to the 8 cells of each strain in 0.1% hydrogen peroxide to induce the oxidative stress onto these different isolates. And then after that, we plated serial dilutions of these cells onto MRS plates and incubated these cells for 72 hours at 30 degrees Celsius. And finally, we observed the colony enumeration and basically the growth of the bacteria visually. And so here you can see just how many. Well, I mean, this is like about a fourth of how many plates we did. So we did a lot. And here specifically is like visually you can see the growth of the bacteria on the plates. Now, as for our results, what we synthesized was that the acidic conditions actually inhibited and slowed the growth of L. plantarum. And also number two, the different isolates, however, showed a diversity of responses to the acidic stress. So as you can see here in this graph, you see a lot of the graphs of the isolates are kind of spaced out and exhibit different responses to the acidic stress. And so, and over here we have the control of the pH 6.5 environment, and so clearly we can see that in the acidic environment, L. plantarum grew slower. But let me make it easier for you though. Right here we have a specific strain of L. plantarum, 1B1. So here is the control group, this pH 6.5, and you can clearly see that initially the growth is clearly more exponential and quicker than the 1B1's growth at a pH of 3.5. And so we found the growth rates of each isolates isolate that we studied, and in every single case, the growth rate at a pH of 6.5 was significantly faster than the growth rate for the isolates at a pH of 3.5. Now, as for the ethanol test, we found out our results were pretty similar. We found out that the presence of ethanol also slowed and inhibited the growth of L. plantarum strains. However, this time, L. plantarum actually exhibited a similar response to the ethanol environment, as you can see through not many gaps between these different isolates. However, the specific strain here, B1.3, is an anomaly and shows clearly significant differences from the rest. And so let's look at AJ11, another isolate of L. plantarum. So here, there's not as strong of a difference between the growths as in the previous acidic graph, but you can still see that in an ethanol environment, AJ11, as well as the rest of this isolates, grow significantly slower than the isolates did in 0% ethanol concentrated environments. And again, we also calculated the growth rates at 8% and the growth rates at 0%. And like the acidic test, every single growth rate for 8% were significantly slower than the growth rates at 0%. And finally, for our oxidative stress test, we found that after 30 minutes of exposure to the 0.1% hydrogen peroxide, most of the L. plantarum isolates died, and their survival was significantly reduced. And so with this graph, we see here that some of the strains were able to grow up until 60 minutes, but we had to extrapolate a lot of information because a lot of the strains couldn't grow past 30 minutes, past the detection limit. And so, what exactly can we conclude from all this data? Well, number one, Lactobacillus plantarum is a very robust species of lactic acid bacteria and can grow in many environmental stresses. And number two, as we saw the diversity of data collected from the acidic stress as well as the ethanol stress, with the acidic stress data in the acidic stress showing many differences. However, contrarily in the ethanol tolerance graph and results, we saw similarities between the responses in the strains. We can deduce from this that Lactobacillus plantarum, through its different phenotypic responses, is genetically diverse. Now, what exactly are the implications to society? For the short-term implication, this will contribute to more research in understanding the idiosyncrasies of the genetic makeup behind Lactobacillus plantarum and lactic acid bacteria. However, in the long term, in the food industry, this research could possibly help fermented food providers ensure healthy products so that they know which environments that their products can sustain in, and so that they can produce the most yield to the market. At this time, I'd like to acknowledge a few people. I'd like to acknowledge Dr. Maria Marco, my principal investigator. I'd like to acknowledge Annabelle Yu, my mentor and my graduate student that I work with every single day. And also, I'd like to acknowledge Rick Pomeroy, the director of the YSP program, Stephen Ramsey, my counselor, the Marco Lab, and my mommy. And I also want to acknowledge you guys for listening to my presentation and putting up with me for 6 weeks.
bacteria for those that face like higher stress?
So for the pH and ethanol tests, we only examined like the growth of the bacteria. However, in the oxidative stress, how we determined the growth was we determined the death of the bacteria. Dave Wayne.
What other stressful environments could you test the bacteria on, like in research?
Like I was saying before, we could test like heat stress and on the different bacteria, as well as like osmotic stress environments. Sean?
What method did you use to like quantify how many cells you had for the oxidative?
It was we just counted them.
Oh.
Yeah,
How much is like a detection?
there was a detection limit because at certain times, like we couldn't really count and quantify how many cells were growing, and so that served as our detection limit. Renee?
Can you give like an example, like which you can use stress or concentration environments correspond to?
So like, for example, for like the acidic environments, when lactic acid bacteria produces or ferments food, it also emits lactic acid. So the lactic acid kills other bacteria living on the food, but it could also be self-destructive and kill itself. So that's why we wanted to test whether or not the acidic environments actually had like a specific effect on the alpine growth. Jack.
I just know that there's one strain that seemed to be really weird in all the graphs. You know anything special about that?
V1.3.
Yeah.
Cosmic. Yeah, V1. So like with V1.3, yeah, like here, it just showed drastic differences. So right now, we think that is because it may have like a different genetic makeup than the rest of the L-plantarum. Isolates. Yes, Renee?
How did you determine what amount of ethanol or hydrogen peroxide to use for your experiment?
We just sort of, I guess, I don't know, we just sort of determined, we just thought that 8% would be fine, and I mean, we just use what goes in the Logan.
Did you replicate this experiment to see if the anomaly occurred again?
No, due to insufficient time. Ellie.
So in the simplified acidic environment graph, the 6.5 pH linear function seems to be stabilized at some point, but the other one keeps on accelerating in the simplified one. But do you think that they will intersect at some point if you tested them for more time? The 3.5 pH linear function and the other one.
Sorry, can you repeat your question, please?
Yeah, so in the simplified, with the one example acidic environment graph that you tested. Next. Yeah, okay, so this one, the 6.5 pH seems to be stabilized at some point, but the one keeps on accelerating at a lower rate, I guess. But do you think that they will intersect at some point if you keep on testing?
Yeah, and probably later on in time, if we still kept testing more than like 70, 71 hours, then they probably would have intersected at some point.
Okay.
Thank you.
Thank you.
All right, here's the Rosenquist
Thank you for listening to my presentation. Now I'd like to introduce Emily Yang, Jack Ustra, and Sabrina Liu to the stage, please.
And we study tyrosine sulfation under
the guidance of Dr. Grace Rosenquist in the Department of Neurobiology, Physiology, and Behavior.
So tyrosine sulfation is a permanent post-translational modification that occurs in the Golgi body of cells. In this process, the amino acid tyrosine, which has a hydroxyl group at the end, has a sulfate group attached to it, and this process is known to increase the binding affinity between proteins. So here's a model that shows you more in detail how tyrosine sulfation works. On the left, you have an unsulfated tyrosine which still has the OH group at the end, and TPST, the enzyme that allows this process to occur, takes a sulfate group from another protein, PAX, and adds it to this hydroxyl group. The extra oxygens branching out from the sulfate group therefore allows for increased binding.
So in 1954, tyrosine sulfation was first discovered in the bovine fibrin peptide B protein. It didn't garner much interest until the 1980s when W.B. Huttner found sulfated tyrosines in ground-up Drosophila. And this sparked a lot more research, which culminated in 1998 when TPST, the sulfating enzyme, was purified from rat liver. And this led to the discovery in 1999 that discovered that a sulfated protein, CCR5, was essential to HIV entry, and without the sulfated component, HIV could not enter into a cell.
So that each of us researches a different aspect of tyrosine sulfation. For Sabrina and I, our goal is to locate potential tyrosine sulfation sites in the proteins that we're studying. And a tyrosine sulfation site is defined as the central tyrosine to be sulfated, and the 5 amino acids to the right of it and to the left of it. To predict these sites, we use a position-specific scoring matrix, also known as a PSSM. So the PSSM looks at the likelihood of tyrosine sulfation in a protein. It assigns a score to each tyrosine site based on how it compares to known sulfated sites and known unsulfated sites. A higher score indicates more similarities with known sulfated sites and therefore a higher degree of tyrosine sulfation. The PSSM that we use is based off of a database of 102 known sulfated sites and 499 known unsulfated sites. It has an operating accuracy of 94.8%.
So specifically, I studied sulfated tyrosine site characteristics. So, like Emily said earlier, a tyrosine site is defined as a sequence of amino acids where you have a central tyrosine and 5 amino acids on either side. And in order to find what characteristics are unique to sulfated sites, I compared a set of 104 confirmed sulfated sites and 674 experimentally confirmed unsulfated sites. Normally, I would compare direct counts in amino acids between these two sets. However, because there's such a large disparity in the sizes of these two sets, I needed to compare the frequencies instead in order to get any easily compared data. So the biggest trend that I found was in negative charges. Negative charges are very strongly correlated with sulfation. The big difference between aspartate and glutamate is this curve that you see right here. Aspartate tends to follow a bell-shaped curve in frequency in sulfated sites, whereas glutamate tends to have just more constantly be high at every position. Even when the spread between aspartate and sulfated and unsulfated sites is the smallest, at the -5 position for the blue aspartate, it still is 5% higher in sulfated sites than in unsulfated sites, which is the entire value of aspartate at the -5 position in unsulfated sites, showing exactly how strong this correlation is. Positive charges are the opposite. Positive charges are inversely correlated with sulfation. The exception here is histidine. Histidine tends to not be correlated either with sulfation and or unsulfation. And this is because histidine in the biological pH where we observe sulfation loses its positive charge. Without histidine, the trend becomes much clearer. Any positive charges that actually do occur are few in number and generally confined to the periphery. Another trend that we observed was in hydrophobics. Hydrophobics show a slight tendency away from sulfation. Now, this varies significantly based on the amino acid being observed and based on the position being observed as well. For example, if we look at the 5 position on unsulfated sites and sulfated sites for phenylalanine, the blue one, and proline, the black one, it is significantly higher in sulfated sites than in unsulfated sites, bucking the overall trend toward unsulfated sites. So although hydrophobics show a large overall trend away from sulfation, it varies wildly depending on what specific amino acid is being examined and what specific position is being examined. So cysteine shows the same inverse correlation with sulfation.
Cysteine, whenever it does appear on sulfated sites, tends to be few in number. This is because cysteine forms disulfide bonds, which makes it difficult for TPST, the sulfating protein, to sulfate the central tyrosine. So the last pattern that we observed in primary sequences is that sulfation is very likely to occur near a terminus, much more likely than not. And these graphs right here show vacancy rates. Sometimes we have the tyrosine site where the central tyrosine is located on, say, the third position of the overall protein structure. That means that the -4th and -5th positions wouldn't have any proteins in those positions. And those are recorded as vacancies here. And as we can see, there's a lot more vacancies in sulfated sites than in unsulfated sites, indicating that sulfation is more likely to happen near the ends of proteins. So with this primary structure data, we moved on to look at 3D, tertiary structure data in proteins and find trends in that. And we were looking for both novel trends, trends that weren't able to be observed in primary structures, and crossover trends, trends that although we did observe in primary structure, it led to both reinforcement of the trends and also maybe some reasoning as to why the trends happened. And we got this 3D tertiary structure data from the RCSB protein database in the form of crystallographic data that we could load into a computer and examine. And the biggest crossover pattern that we observed was in the negative charge residues and the proximity to terminus. If you see here on this figure, we have the sulfate tyrosines in yellow, the negative residues in blue, and the terminus circled in red. The negatively charged residues create an electrostatically negative area in space and the positively charged to attract the positively charged TPST, which induces sulfation in the nearby tyrosines. Additionally, the proximity of the tyrosines to the terminus indicates that the site is near the surface of the protein, making it easier for TPST to come in and sulfate it because it's near the top. A more surprising and novel observation that was made from this tertiary structure information is that hydrogen bonds form with the sulfate group on sulfated tyrosines with histidine, serine, threonine, and other positively charged amino acids. Here, we see a human P-selected in complex with the PSGO1 glycosylpeptide, and hydrogen bonds being formed between the sulfate group and the serines and the histidines. This was experimentally confirmed in crystallography data over here with the same protein complex, and really gets to the core of what tyrosine sulfation does. It increases this hydrogen bonding network right here, which increases the overall rigidity and the overall adhesion of the complex. This, again, also happens with lysine. Here we have a thrombin inhibitor's lysine forming a hydrogen bond with a pyridine variant 2's sulfated tyrosine. It also happens with arginine, although to a much less extent than lysine, about 5 times as less. So although you will see arginine forming hydrogen bonds with the sulfate group on sulfated tyrosines, it will just happen less. So again, the main effect of tyrosine sulfation is this increase in the hydrogen bonding network, which increases the rigidity of the overall complex between the proteins and increases the adhesion between the proteins that are interacting through tyrosine sulfation. And hopefully, the patterns that were observed from the primary structure analysis will add a qualitative element to what has hitherto been a quantitative prediction of tyrosine sulfation in the form of the PSSN. Thank you. Any questions?
Why did you specifically have a range of 5 amino acids on each side?
So 5 amino acids gave us enough data to be able to look at statistically significant trends. And past studies have used this site defined by 11 amino acids with a central tyrosine and 5 on each side. So we just kind of stuck with that.
Just a quick question. Do you know what program you guys use to view the crystallography data?
Yeah, we use PyMol. And alternatively, if you want to take a more bioinformatics approach, there are Python libraries to be able to parse them and analyze structures for it. I did that too when I was looking for the hydrogen bonds.
You talked about an increase in the rigidity. Did that compare or affect the function of the protein?
Absolutely. When we were talking about the earlier when HIV, it was discovered that tyrosine sulfation was essential for HIV. That was because the adhesion between the HIV and the receptor wasn't strong enough if you remove the sulfate group. So those hydrogen bonds can make a significant difference when you're looking at protein adhesion, protein rigidity.
In what types of cells is tyrosine sulfation more likely to occur?
Receptors. These guys have done most of the research on it, and we'll be talking more about it. Any other questions? No? All right, well, then I'd like to introduce Emily Yang.
Hello. So my research project focused on predicting potential tyrosine sulfation sites in coherent proteins. The coherent protein family is a large and diverse superfamily of transmembrane glycoproteins. And a glycoprotein consists of a polypeptide binding to carbohydrates. Different coherents are found in different parts of the body, where they have specific functions ranging from cell movement, boundary formation, and cell recognition. Problems in coherent expression have even been linked to cancer development, which goes to show how important these coherents are to our bodies. So here's a diagram of a classical coherent. The classical coherent is a coherent subfamily consisting of the type 1 coherents and the type 2 coherents. A classical coherent is characterized by the 5 EC repeats in the extracellular region of the protein. They are also highly dependent on calcium ion binding. So when a calcium ion binds to the coherent, it causes a change in structure, which then influences how that coherent interacts with other proteins, especially other coherents. So 3 calcium ions bind to each negatively charged junction between the EC repeats. So in a classical coherent, we have a total of 12 calcium ions binding to one protein. Most coherent interactions are homotypic, meaning that one coherent type interacts with a coherent of the same type. This type of interaction is crucial for germline formation during embryo development, as well as tissue maintenance and formation. So initially, I looked at 20 coherents from various coherent subfamilies. However, in my PSSM results, I found that the most interesting data came from the type 2 coherents. So type 2 coherents are expressed in various body tissues, although a majority of them are found in the central nervous system in places like the brain. Some of their specific functions include embryonic development, axon sorting, and regulating certain brain functions. So out of the 11 type 2 coherents in this study, I was able to identify 6 repeated potential tyrosine sulfation sites, which I mapped out here. Some of the more interesting sites are this bright pink site, this green site, and this salmon pink site. Each of these 3 sites are located in the negatively charged calcium binding region of the coherent. So initially, I thought that tyrosine sulfation could have an effect on calcium binding in the coherent. Of these 6 sites, the most interesting data came from this green site with the ADDP motif. For the remainder of my presentation, I will be focusing on this site. So this site was highly conserved, and conservation of a site indicates that it's being repeated in various proteins and ideally in different species. This is significant because preservation of a site over time in natural selection indicates that it must have an important biological function in the organism. So this site was highly conserved as it was found in 10 out of 11 type 2 coherents. If you look at the site sequence here, you'll also notice that the sequence is highly conserved with at most 2 differences in amino acids. Also interesting is that a majority of these sites have a PSSM score that is really high. And a high PSSM score, as I mentioned earlier, indicates higher chances of tyrosine sulfation. Another interesting point about the site are these 2 negatively charged aspartate amino acids located near the central tyrosine to be sulfated. So tyrosine sulfation is more likely to occur in negatively charged sites because the sulfating enzyme TPST is positively charged and will therefore more easily bind to negatively charged regions. So the concentration of this negative charge near the tyrosine is conducive to sulfation. I then looked at this site in a 3D model and found that the orientation of the tyrosine supports sulfation. And these images will be better seen in the dark, so can I get the lights off?
Mm-hmm.
Thank you. So on the left here, we see a coherent 6 in a diamond formation. So that's two coherents sticking together. In its monomer form, this coherent 6 would display the predicted tyrosine site, highlighted in pink, on the surface of the protein. This is significant because the tyrosine would be more exposed to the sulfating enzyme TPST and therefore more likely to be sulfated. On the right here, we see a close-up of the tyrosine to be sulfated, which is colored in red. The hydroxyl end of this tyrosine is jutting out of the protein. And this is significant because the sulfate group is added to the hydroxyl end of the tyrosine. And if the tyrosine is pointing away from the protein, then it's more accessible by TPST, the sulfating enzyme, which can easily attach the sulfate group and sulfate the tyrosine. This site is also located in the negatively charged calcium binding region between EC1 and EC2. Colored in yellow here are the negatively charged amino acids in this region, while colored in red is the predicted tyrosine sulfation site. As I mentioned, tyrosine sulfation is more likely to occur in negatively charged sites, and the fact that this site is located in a negatively charged region increases the likelihood of the chances of tyrosine sulfation. I then looked at the site in various species, including humans, mice, rats, and bovines. And interestingly, I found that the site sequence was completely conserved in these species for a coherent 6 protein. And a higher degree of conservation indicates biological significance, which could be connected to the post-translational modification of tyrosine sulfation. In conclusion, I have reason to believe that this site is a potential tyrosine sulfation site. It has high PSSM scores, it is conserved in the type 2 coherence, and it is conserved in different species. As well, its positioning in a 3D model also supports sulfation. An important characteristic about this site is that it contains a known calcium binding site. So initially, I thought that tyrosine sulfation could increase calcium ion binding, as previous studies have shown that tyrosine sulfation increases certain metal ion binding. However, recently I read a paper demonstrating that tyrosine sulfation actually decreases calcium ion binding in a particular peptide hormone. And a peptide hormone is basically a protein that acts as a hormone. So if this study is true for all proteins, then this site is probably not an actual tyrosine sulfation site. The main reason is that calcium ion binding is so crucial to coherent function and structure that this site would be an evolutionary disadvantage to the organism and probably be discarded over time through natural selection. However, not a lot of research has been done on tyrosine sulfation on calcium ion binding, especially in glycoproteins, which is what the coherent is. And I also want to mention that the study was done on a peptide hormone rather than a glycoprotein. So it's possible that the effects of tyrosine sulfation on calcium ion binding is dependent on the particular composition and structure of the site or the particular protein. It's also interesting that 6 of the repeated potential tyrosine sulfation sites I identified were located in the calcium binding regions. So, in conclusion, more research needs to be done to clarify the effects of tyrosine sulfation on calcium ion binding, as well as to confirm the presence and location of tyrosine sulfation in the coherent protein. In doing so, we'll be able to deepen our understanding of coherent structure and function, and hopefully one day we'll be able to apply this understanding towards developing new ways of targeting coherent-related diseases, such as cancer. Thank you very much. That concludes my presentation, and I'll take any questions you have for me.
So you said that coherent 6 was very highly conserved across a lot of other species. Were the other coherents that you saw that had this site also highly conserved as well?
They were highly conserved, like coherent 7 was highly conserved. I wasn't able to find sequences for all the coherents, but the ones I could find were pretty well conserved.
Okay, so would you guess that all of this family would be highly conserved across like a variety of species then?
That's what I hope so,
Yeah.
but I can't make any assumptions.
Okay.
How do you distinguish a high PSSM score?
So scores higher than 1 we consider, and then scores higher 2.42 we highly consider. And the reasoning behind this is that 2.42 is the score where the chances of a false positive and a false negative are equal. And a false positive is where a site is predicted to be sulfated, but it's actually not. And so we look for scores higher than 2.42 to reduce those false positives, as well higher scores indicate more similarities with known sulfated sites, which indicates a higher chance of sulfation.
What's the highest score you can get?
I'm not sure. But the highest score that I was able to get was around 5. But Sabrina had higher scores. So, yeah.
You really briefly mentioned that studying coherence is related to cancer development.
Well, certain coherents have been connected to regulating how a tumor forms, and there's not a lot of research being done on that, but hopefully more research on tyrosine sulfation will be able to develop new methods on cancer treatments. And obviously more research will have to be done on how coherence regulate cancer development as well. Okay. I would now like to introduce my friend and lab partner, Sabrina Liu.
So for my project, I'm looking at the potential of tyrosine sulfation in voltage-gated sodium channels. A voltage-gated sodium channel is an ion channel that regulates the sodium intake of a cell based off electric current traveling through the cell membrane. Here you can see a model that shows the linear form of such a channel. As you can see, there are 4 domains, and in reality, these domains are arranged in a clockwise fashion to form a 3D cylinder that has an actual pore in the membrane. In each of these domains, there are 6 transmembrane segments, known as S1 to S6 from left to right. On the very far right of each of these domains, you see 2 segments, S5 and S6, and these 2 segments line the inside of the pore and can therefore easily come into contact with various substances passing by. Between S5 and S6, you see this extracellular reentrant loop, and that is known as a P-loop. In the 3D structure, it is obvious that the P-loop kind of juts out of the pore and in towards the center, and this is because the P-loop regulates sodium permeability. Now, there are many ways to inactivate the sodium channel. The way that we're most interested in is using the positively charged S4 segment in each of these 4 domains. In order to balance out the positive charge located in this segment, the regions from S1 to S3 have to be negatively charged. So voltage-gated sodium channels are found all throughout our central nervous system, such as in our brain. In addition, they're found in peripheral nervous system, as well as skeletal muscle tissue, like in our appendages, as well as cardiac myocytes, like in our heart. All in all, there are 10 different voltage-gated sodium channels located throughout the human body. However, one of them, NAV 2.1, is evolutionarily very distinct from the others and lacks strong homology. Therefore, for purposes of this research, data from NAV 2.1 has been omitted. All of the other 9 channels, however, are evolutionarily very similar, especially these 3, NAV 1.1, 1.2, and 1.3. The close proximity of these 3 sites would suggest to us that we should be able to find some sites that are conserved among these proteins, or sites that are important to cell function that have been saved through evolution. And indeed, we do find such sites. Here are 3 potential tyrosine sulfation sites, all with pretty high PSSM scores that have been conserved among all 3 of these proteins. And as you can see, they at most have 2 amino acid difference. These 3 sites are scattered all throughout the voltage-gated sodium channel. The blue site, site 1, is located on the P-loop of domain 1. Now, as I mentioned before, the P-loop kind of juts out from the rest of the channel, making it more easily accessible by sulfating enzymes. The second site, site 2, the pink site, is located on the extracellular loop between S1 and S2 in domain 2. So as I mentioned before, this area is negatively charged. TPST, the enzyme that allows tyrosine sulfation to occur, is typically attracted to negative amino acids, further supporting sulfation at this site. Our last site, site 3, the green site, is located on the tip of S6 in domain 3. And like site 1, therefore it is able to easily come into contact with sulfating enzymes. For this evidence, we will focus on for this project, we will focus on evidence that supports tyrosine sulfation in site 1 and site 2. The first piece of evidence that supports tyrosine sulfation in site 1 is homology. Of 9 of the voltage-gated sodium channels that we sampled, 6 of them have a conserved site at this position. And in fact, they all have pretty high PSSM scores. We see that among these sites, there's at most 1 amino acid difference. And the 1 amino acid variation in NAV 1.7 causes the PSSM score to jump way up to 7.15. the highest PSSM score of any potential tyrosine sulfation site among the voltage-gated sodium channels. These high PSSM scores indicate that these sites are likely to be sulfated, and the fact that they're so well conserved indicates how important it is to channel function. Therefore, anytime that we see a dramatic mutation in the amino acid of these channels, we'd expect to see some kind of cellular malfunction. And there are actually mutations in this site that do that. In this mutation, the threonine in the positive 3 position of this site, which is highlighted in orange, has been mutated to arginine, and it causes the PSSM score to decrease by over 2-fold. In addition, patients with this mutation experience epilepsy. Now, this mutation was found in NAV 1.1, a voltage-gated sodium channel that is located in the neurons of our brain and is known to regulate signal transport. Therefore, the relationship between these two effects, the fact that the PSSM score drops so much after mutation, meaning that after mutation, the site is very unlikely to be sulfated, in addition with the fact that these patients suffer from epileptic seizures, shows us that perhaps tyrosine sulfation plays a crucial role in these channels in monitoring signal transport. In addition, we have similar evidence that supports tyrosine sulfation in site 2. Now, site 2 is located in the extracellular region between S1 and S2, and we already know that this region is related to FASN activation. Now, in order for scientists to narrow in on exactly which amino acids are involved, they had to scan through the site, mutating each amino acid as they go, to figure out which one has a dramatic effect. Now, when they mutated the glutamate, the E, and the -2 position to a K, a lysine, they found that it slowed down the FASN activation of this channel. In addition, glutamate is a negatively charged amino acid, while glycine is a positively charged amino acid. TPST, the enzyme that allows tyrosine sulfation to occur, doesn't typically bind to positive amino acids, which explains why the PSSM score drops so much. It goes from a positive score, meaning the original unmutated site is actually more similar to known sulfated sites, while after being mutated, the negative score indicates that this new site is more similar to known unsulfated sites. The relationship between these two effects further supports that tyrosine sulfation plays a role in signal transport. In addition, we have evidence that toxins actually bind to this site, known as a beta-scorpion toxin. Once again, scientists use a similar method, scanning through the site and mutating amino acids as they go, to figure out exactly in which region the toxins bind. Once again, they found that the same glutamate in the -2 position of the site, when mutated to a cysteine, actually reduces toxin binding by 3.5 to 5.6-fold. In addition, we see the similar drop in PSSM score from a positive score to a negative score. Now, we already know that tyrosine sulfation is responsible for a greater affinity of protein binding. So when we see that a mutation like this causes the PSSM score to drop so much, as well as drops the binding of toxins so much, it leads us to believe that tyrosine sulfation not only occurs at this site, but could play a role in the binding of these toxic proteins. Therefore, in conclusion, we have strong evidence to believe that site 1 and site 2 are both likely tyrosine sulfation sites. Not only do they have high PSSM scores, but they are also conserved between many of the channels that we sampled. In addition, if these two sites were to be sulfated, we have mutagenesis evidence that shows what would happen if they're mutated such that they could no longer be sulfated, leading to cellular malfunction. Finally, for site 2, we have evidence that toxins bind to this site. And because proteins bind to the site, it is characteristic of a site that is tyrosine sulfated. So what are the implications of this research? Now, I already showed you that NAV 1.1 and other channels located in the neurons of our brain have mutations associated with them that are linked to epilepsy, such as syndromes like Dravet syndrome. In addition, other sites like NA1 and NAV 1.5 that are found in cardiac myocytes have mutations on them that are associated with arrhythmia, which is when your heart slowly starts to beat very fast and very irregularly and can lead to sudden death if prolonged for several minutes. Now, there are already drugs out there that target voltage-gated sodium channels. However, there are no confirmed tyrosine sulfation sites on any ion channels thus far. Therefore, more research needs to go not only finding more sites like the one on NAV 1.5, but also confirming that the two sites I showed you today, site 1 and site 2, are actually tyrosine sulfated. Thank you for listening to my presentation. So at this time, we would like to acknowledge all the people who have helped us in our research. First of all, Dr. Grace Rosenquist for her mentorship, as well as Yan Nguyen, Malonzo, and Vineet Gangaram for their valuable discussion. In addition, we would like to thank all of YSP for giving us this opportunity. So now we'll take any questions. Yeah, Michelle.
So how does the positive charge in the S4 signal aid in fast methylation?
Good. So like as I mentioned earlier, there's electric current traveling through the cell membrane, and it's basically regulating how much sodium the cell is taking in. So what happens is the voltage kind of builds up at this site, and previous evidence show that the site would actually kind of screw shut, but recent crystallographic data has shown that it actually acts more as a paddle, and that actually further supports that tyrosine sulfation could happen there, and further supports that toxins could bind there. Eileen?
So why is it that the mutation in cysteine lead to such a huge decrease in the PSS score?
Okay, so I guess this is a place where we can see how Jack's work kind of applies.
If you'll remember back to my slide, cysteine is very inversely correlated with sulfation. So that is reflected in the PSSM score with a sharp decrease.
Yeah, so the fact that like both of our projects kind of match up is kind of cool. Logan?
What steps do you need to take to confirm your tyrosine sulfation site?
So I think the experimental data need to confirm tyrosine sulfation is actually very complicated. That's why, as of right now, we only have like 104 confirmed tyrosine sulfation
Right,
sites.
you need to take each protein and do X-ray crystallography on it. And then, only then can you actually get the sites with like several angstroms of precision and like actually get like where the sulfate site is. But without that, we don't have any current way of doing it.
I guess that's why so much work needs to go into predicting these sites, so we actually have some leads and we're not just wasting resources. Okay. Ben?
So you talked about the relationship between tyrosine sulfation and calcium binding? Yeah, sorry. So you said calcium is crucial for some sort of protein function.
Yeah.
What kind of effects has tyrosine sulfation had or been found to influence protein synthesis that could, since they all seem to be correlated? You said there's a lot of high PSSM scores near calcium binding sites.
Yeah.
So is there do you have any idea why?
Why there are high PSSM scores?
Yeah.
Well, part of the reason could be that in calcium binding sites you have a lot of negative charge, and known tyrosine sulfation sites are also highly negatively charged. So there could be a correlation there. And it could be that the fact that a tyrosine is located in that negative region just happens to give us a site with a high PSSM score. So it could be that when a site shows up in the PSSM, it's not necessarily due to sulfation, but due to its location in that region.
So do we know if there are any positive effects of tyrosine sulfation that could counteract the negative effects of losing the calcium binding?
Well, tyrosine sulfation hasn't been confirmed in coherence yet, and that's why we're predicting right now.
Any other questions? No. Okay, thank you for listening to our presentation.
There's going to be a 15-minute break right now.
Actually, ladies and gents, we have a more significant break. We have
M1 and M2 macrophages in domestic rats. This research was conducted at the Center for Health and the Environment under Professor Dr. Kent Pinkerton and lab supervisor Dale Uinami. Smoking touches the lives of countless people in the United States. Currently, 17 out of 100 adults aged 18 or older smoke cigarettes. This means that there are over 40 million people who are impacted by tobacco in the United States alone. Active smoking is closely linked with chronic obstructive pulmonary disease, also known as COPD. COPD encompasses the symptoms of both emphysema and bronchitis. These patients face irreversible damage to their lungs, including broken down alveolar walls, which hinders airflow, as well as increased cough and sputum production. COPD is the third leading cause of death in the United States, and the mortality rate is on the rise. Even though the effects of tobacco are widespread, there are few treatments for those who are suffering. One of the larger goals of my project is to determine the cellular changes that happen in the lungs and use this research to develop medications and also to contribute to the greater cause of convincing current smokers to quit before any further damage can be done. My research is intended to wrap up a long and ongoing study on former smokers. In previous research, significant differences were found between the filtered air rats and the tobacco smoke rats. These differences included increased epithelial thickness, which is the tissues lining the lungs, increased mucin content, which is mucus, as well as a decreased amount of tunica adventitia, which is the outermost layer of the artery. In a study done similar to mine in my lab, it was conducted on current smoker rats, and there were significant differences shown between the numbers and phenotypes of the macrophages between the filtered air groups and the tobacco smoke groups. This led to a curiosity on whether a period of smoking cessation would affect these numbers and phenotypes. The purpose of my study is to determine the effects of smoking and quitting, specifically what the effects are on the macrophage numbers and phenotypes. Now, to get more depth about macrophages themselves, a lot of people like to compare macrophages to Pac-Man, and this is because they both engulf things. Macrophages engulf things such as cellular debris and foreign substances, basically anything without the healthy human body marker on it. Macrophages can be split into two groups, M1 and M2. The M1 phenotype macrophage inhibits cell proliferation and therefore leads to tissue damage, whereas M2 macrophages do the exact opposite. M2 macrophages encourage cell proliferation, which therefore leads to tissue repair. These differences are important to note because they give us information on where the lung's at in the healing process. For example, if we were to find an excess of M2 macrophages in the cell, we would conclude that there was healing being done. It's also important to note that these macrophages are in old rats, so there might be an increased number. So all research in the study was done on spontaneously hypertensive rats, known as SH rats. There were 74 of them in this study. 38 were in the control group, and 36 were exposed to tobacco smoke. Well, we chose spontaneously hypertensive rats because they are proven to show symptoms of COPD and have increased susceptibility to airway diseases. So these rats were exposed for 6 hours per day, 3 days per week, at the age of 2 months and 2 weeks old. They were put in these smoke chambers right here and exposed to research-grade cigarettes made at the University of Kentucky. After 39 weeks, all exposure stopped. These rats were allowed to live the rest of their lives with no additional exposure. So what this means is that these rats were meant to imitate former smoker rats. Out of the 74 rats, 12 of these rats' tissues were chosen for further analysis. These rat tissues were chosen randomly, and 6 were from the spontaneous, 6 were from the filtered air group, and 6 were from the tobacco smoke group. So there was 2 types of staining done on these lung tissues. The first was hematoxylin and neopene staining, also known as H&E. This type of staining stains the nuclei blue and the surrounding tissues in the cytoplasm pink. Now, this is a type of non-specific staining that gives a general overview of the cell but does not allow for the differentiation between the two phenotypes. So this is what an H&E stained micrograph looks like. And right here is a macrophage, and you can tell it's a macrophage because there's pink and then a blue nuclei, and they reside in the alveoli of the lungs, which is the airways, air spaces. The second type of staining done was immunohistochemistry, known as IHC. So this type of staining is much more labor-bearing, and it takes around 6 hours. But it also allows for the differentiation between M1 and M2 macrophages. So first, the primary antibody connects to the antigen of choice, in this case M1 or M2, and it's left to sit there for an hour. Then the secondary antibody is added, and this connects to the primary antibody, and this antibody is important because it contains an enzyme substrate complex that actually colors the antigen of choice brown. So these are photos of M1 stained tissues and M2 stained tissues. So a positive macrophage would be counted if it's brown with a blue nuclei as seen right here. The dilutions chosen for these stains were 1 to 50 and 1 to 2000. And this was in order to get the best color payoff. After all staining was done, work was done with a Zeiss microscope. So 20 photos were taken of each field, of each slide. These were 20 random photos of each field, and this was to get a general overview of everything in the cell. So before I get into my results, I would like to go over some lung anatomy. And this is what the tissue looks like in stained slides. And so over here is the subpleural region, and this was one of the first regions that clusters of macrophages were found. And also shown in this picture are the airways. On the other picture, there's a terminal bronchiole right here, and seen right here are another area of clusters of macrophages. So the first type of conclusion drawn was through qualitative assessment. And basically we concluded that there are more macrophages in the tobacco smoke animals when compared to the filtered air animals. And before I even began my research, I was asked to determine whether lung tissue was a smoker rat or a filtered air rat. And before even beginning any research, I pinned 5 out of the 6 correctly, and that's because the difference in macrophage numbers is very apparent. Quantitative research was also done with these rats, and significance was found between the filtered air and tobacco smoke animals pertaining to the M1 macrophage. What this means was that there was a statistically significant difference found between these two groups. So in a greater context, this means that there's still inflammation in the lung after a period of smoking cessation. There was no significance found between the differences in numbers of the M2 macrophage, however. This means that even after a period of smoking cessation, there was no effective repair in the lungs. The third conclusion drawn from my project was that macrophages could be in transition. What this means is that macrophages could be both M1 and M2. At first, this was thought to be an experimental error because it was thought that macrophages would either stand for one or the other. But upon further research, it was discovered that outside signals could cause the macrophages to change phenotypes. Another thing I'd like to note is that macrophages in the tobacco smoke animals appear to be really foamy and enlarged, and we hypothesize that this is because these macrophages were engulfing lots of things. The final conclusion drawn from my research is that there's persistent inflammation in the lungs even after a period of smoking cessation. What this means is that former smokers never truly fully recover. Although there has been significant research done in this field, there's still so much more to discover. As stated earlier, medications is a large part of this research goals. We hope to continue on and determine the specific pathways that lead to inflammation and help create a more targeted form of medication. We would also like to determine the specific inflammatory mediators that are released from these macrophages that cause the fight or fix response. So I'd like to thank my professor, Dr. Kent Pinkerton, as well as graduate students Alexa Pham and Zing Zing for guiding me through my research. I'd also like to thank everyone at the Center for Health and the Environment for giving me not only a research experience, but memories as well. And then I would like to thank all of you guys for experiencing this with me. Thank you. Yes.
You mentioned how you used spontaneously hypersensitive rats for the experiment. You mentioned how they were more susceptible to diseases, but are there any other differences from compared to normal rats?
Well, we chose these rats for this study because we knew that they were more susceptible to these airway diseases, and that was one of the main reasons we chose them. Yes.
How did you know for sure that their macrophages were positive and not just negative?
Well, in the M1 and M2 stain slides, there was a negative control, and this negative control wasn't stained with a primary antibody, and therefore there should have been no staining on that slide. So if we were wondering whether a macrophage was a positive macrophage or not, we could refer back to the negative control, and if that area in the same section of the lung was clear, then we could conclude that it was a positive macrophage. David?
How would you use your research to convince current smokers to stop quitting? Because, like, basically what you're saying is that the effects of smoking, like, never, like, cease.
Well, in my paper actually, I wrote about how even though there are still symptoms of smoking, there is like pluses in quitting, and one of these I found was in mental health. These smokers no longer face depression-like symptoms. So yeah. Kane.
So did the error bars in the M1 and M2 macrophage charts, did they like overlap within the chart? It would seem like some of them were pretty big.
Oh, like, so did the like black and white graph?
Yeah.
That is a potential because they are stained for both M1 and M2, but that means that they possess that phenotype, so then they could be counted as a positive macrophage.
How did you determine the length to expose the rats to smoke?
Well, the length is determined, so if we extrapolate the ages of the rats, these rats started exposure at 2 months and 2 weeks, which is the equivalent of the later teen years. And then 39 weeks later, it'd be an equivalent of an adult human smoker around 35. And that's how we decided. Any other questions?
What was the M2 macrophage supposed to be doing in the lung?
One more time?
What was the M2 macrophage doing in the lung as it was supposed to be healing?
Yeah. Come again?
You said that the lung is never healed completely, but what does the M2 macrophage heal?
Well, it's healing the tissues, so it's repairing the tissues. But since we didn't find a lot of them, we concluded that there wasn't a lot of healing being done. Any other questions? I would like to invite my friend Delaney Buscard up here. I'm sorry if I start coughing. There's now.
Hi. Good afternoon, everybody. My name is Delaney Buscard, and for the past 6 weeks, I have been studying how surface charges on nanoparticles influence the deposition and retention of the nanoparticles inside the respiratory systems of mice. I've been doing this at the Center for Health and the Environment under Dr. Kent Pinkerton and Ariel Parker. So, respiratory diseases rank among the top 10 causes of death in the world. COPD is ranked 3rd, followed by lower respiratory infection, and later various lung cancers, as seen in Figure 1. So nanomedicine has been on the rise as a treatment for this because of its versatility and interdisciplinary nature. For example, nanoparticles can be used for early disease detection and drug delivery. So, mesoporous silica nanoparticles are the particles that were being worked with in this study. Figures 2A and 2B show these nanoparticles. A A being the negative ones and B being the positive ones. Now, they're not too different in size, but just a little bit. They are porous, as you can see from the name here, and their pores can be filled with medicinal compounds, which can be used to target the respiratory system specifically, and hopefully even the entire body. Now, characteristics of a good drug delivery system overall include large pore size, high absorption of drugs, site or cell directing ability, so like being able to control what cells the particles go into, and a timed release. So the purpose of this study was to determine what these MSNs do after being deposited into the respiratory tracts of mice after a single exposure for about 5.5 hours. Another further analysis also pointed to if surface charge, so if either of these nanoparticles had different retention rates. So past research has indicated that MSNs are safe for acute exposure since they didn't cause any inflammatory response and the respiratory system remained unchanged in terms of like anatomy and the cells. However, this research did not point to long-term consequences, and it didn't indicate if charge had any impact. The actual method that was used during the study was aerosolization. So a solution was run through here, the chamber, and here are the mice, and here's the nebulizer with the aerosolized MSNs inside. Now, this method actually did maintain the structural integrity of the MSNs, so hopefully we can use them for future research. So mice were used in this study because they are good human models. They can develop many of the same diseases that humans have, especially those of the respiratory system. In this study, CD1 male, 8-week-old mice were used, and they were exposed to 5.5 hours of aerosolized MSNs, both positive and negative, and after which they were sacrificed 0, 1, 7, and 21 days post-exposure. So, after inhalation and after being sacrificed, their lungs were lavaged, which means they were flushed out with phosphate buffered saline, or PBS, to collect the loose cells. Then that valve was used to prepare cytosine slides, and that was stained with DAPI and imaged under a confocal microscope. Now, the actual lungs themselves were sectioned and embedded in paraffin, and then later stained by me with H&E, hematoxylin neosin, or DAPI, as seen in these two figures over here. Then those DAPI stained sections were imaged at the CAMI core facility here and here at UC Davis out at CHE. So the first analysis that was done was using the cytospin slides. I looked at images that were taken by the graduate student, Ariel Parker, and we were able to determine if macrophages had MSNs or not. If they didn't have them, then that would be in the count. So overall, the calculations for this graph were the total number of macrophages the number of macrophages with MSNs were divided by the total number and multiplied by 100. So this graph shows them as a percentage. The blue bars represent the negatively charged nanoparticles, and the orange bars represent the positively charged nanoparticles. So as you can see in this graph, the orange ones kind of have almost a constant rate, and there's a decrease within the negative time points. No statistical significance was found for these data. So, aerosol characterization. So during the actual exposure, a cascade impactor, which is right here, was used to sample the aerosols and characterize them based on size and distribution, since the idea was that they're inside a droplet of nanopure water, and once that water is in the respiratory system, it explodes because, yeah. And then the nanoparticles are released into pollution. So figures 11A and 11B show the dried aerosols. So after 1 week after the exposure, they're weighed, so we can talk about particle distribution. And C and D show one aerosol droplet with multiple MSNs inside. So this graph used gravimetric analysis to characterize the aerosols. Bless you. So for this study, we only had time to run the negative particles, and it was determined that the median diameter was about 1.5, and that kind of makes sense based on the output of the nebulizer that was used to administer the aerosols. Okay, so now we're going to turn off the lights so we can talk about fluorescence. Everybody see it? Okay. All right, so figures 13 and 14 both show lung tissue. 13 is a collapsed lung from 1 day post-exposure to negative MSNs. These bright spots over here are the MSNs, and we can conclude that they are in the airways. Over here we have non-collapsed tissue from 21 days, but only exposure to filter air since this was part of the control. Bless you. And some of this is also just autofluorescence, because of blood autofluorescence. Okay, so another conclusion that can be made based on the first graph is that the negatively charged nanoparticles are most likely being released by the macrophages that engulf them due to that decrease in the bars, and the macrophages are retaining the positive ones. Another conclusion is that based on the H&E stained sections, there was no inflammatory response to these MSNs. Further research can also determine what percent the MSNs take up of the cell, and also how long they last within the respiratory system. So ongoing research. These are the cytosine slides that I was analyzing throughout the summer. The blue are nuclei, and the red over here and stuff are the MSNs. So first I would determine if it had MSNs or not, and if it did, then I would calculate CTCF, which stands for corrected total cell fluorescence. Now, that was used to quantify the MSNs, so you can see that this one isn't really intense, there's like a couple, and over here there's a lot, and we wanted a lot to be more quantified. So a cell was selected, and its area and integrated density were calculated, and then an area of background with little to no fluorescence was selected, and its integrated density and area were also calculated. Those two were compared, and based on that we were able to create ranges of CTCF, and we came up with the frequency of how many cells are within each of those ranges. Now, from preliminary results, which are not valid for presentation, I was able to determine that T1, so 1 day post-exposure, was the only time point for both positive and negative MSNs that had one with the highest level of CTCF, which was somewhere in the millions. Now, that indicates that most likely the MSNs kind of clump in the beginning, and then over time they declump and move around a little bit. So these are the references that I used for my research and my acknowledgements. I would like to thank everybody who has helped me do this. I know this has been a stressful process for everybody. I would especially like to thank Dr. Pinkerton and Ariel. Ariel cannot be here today. And also the staff at CHE for having fun and eating cookies. Also Corinne for driving me out to lab. And then Dr. Pomeroy and Megan for being so supportive and everybody. So, questions?
So there's no fluorescent side, change the properties of the MSN?
No, it's actually, so in the process of synthesizing the MSNs, it's one of the dialite is actually one of the last things added, and it just coats it. So it just makes it easier for us to see it under the microscope. It doesn't change anything. Alexandra?
So when you talk about your histological method slide, why did you do H&E and DAPI staining?
So H&E was just to see if there was any inflammation, and we wanted to you can't exactly cell differentiate with H&E, but you can tell between inflammatory and non-inflammatory cells easily. And with DAPI, it's a counter stain to the dialite. The dialite was red and the DAPI was blue. And we were able to see if a cell had MSN or not. Rebecca?
So you talked about how the respiratory system mice are similar to humans, and so you said in your experiment the exposure time to the MSNs was about 5.5 hours. So I was just wondering what that equivalent would be in humans.
It's about 4 minutes in humans. Ben?
So you said that the particles were being absorbed by macrophages in central lung tissue. Does that affect medicine?
Well, when we did the study, they're empty.
Okay.
So hopefully, when we do eventually put medicine inside, they are capped off, and that cap off is more organic, and it can just come off at a certain time once it reaches its desired site. So let's say we wanted them to only go to respiratory cells, and once they're in the respiratory system, it would cap off and then release the drugs.
And the macrophage would eat it and there would kind of be no loss.
Is that it? I would like to introduce Sunny Hui.
Sunny, we're a few minutes ahead. Is everybody who was planning on being here here?
No, not yet.
Okay, then we'll wait.
Okay.
We're like Amtrak. We're off. We're never early.
Each of your girls individually.
I created, oh, I did.
Okay, good.
But then I didn't create
You brought what's
the
on the
return to the top.
Okay. I thought you were doing that in like all of them.
No, I made,
Leave your painting.
right, and you know what?
Did you do it?
Half of the people didn't work. Half of them didn't work. Half of them went to the So,
Okay, I get it.
I'm reading these things on Sunday night at 9 o'clock and trying to stay awake. It's got to be boom, 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 boom. Screws up, 0 credit. Yeah, but I created it, it's my own fault.
Okay, just say hi to me.
Besides, it's the kind of ridiculous stuff I can
I'm trying to figure this out.
do. So I send a message to Kelly.
That I
Did I show you the picture?
No, you didn't show it to me. You said it was ancient. Perfect
One of these guys, the guy from
Pedro, he does pick up.
well, I knew that.
She
But
stays strong all the time.
did she? Despite this phone call?
According to her, or the last time I checked, she did.
We can't have one more of these.
Are they here or no?
They're not here.
Okay.
Are they coming? Yeah, we'll give them a couple.
Do you like text them?
Is this like, so it's not your parents anywhere? So Stephen, the other thing about I doing it this way, I can do the first one, when they do the left one, that's the way it's supposed to work. Now you
Yeah.
but I'm not sure.
We have them all tomorrow. I hope you guys do that when I was here. But I looked at it like, they're all day.
It's like it's at what
Just glancing around.
Oh, I know.
They went to the Young Center.
Oh, yeah, they were on my side. Are they there now?
Yeah.
Okay.
Is that far?
Did they, yes, they walk, drive.
That's a good sign.
Is this your lab?
Yeah.
Yeah.
Say that to everyone.
We're just working into a break. We're just
I know.
So, why don't we give them a break?
Yeah. Okay. Alright, ladies and gents, we're gonna go ahead and take our break early, and Sunny's gonna come back and do his presentation at about 3:45, between 3:45 and 3:50. So can you be back in your seats by 3:45, please? Yes. Okay, thank you.
And don't make too much noise in the hallway.
Why do you
supervision of graduate student Jennifer Leanne in the UC Davis Department of Chemistry. For the past 6 weeks, I've been investigating the mechanism behind chemical enhancement of gold nanoparticles under X-ray irradiation through electron paramagnetic resonance. First off, I will introduce nanoparticles. Nanoparticles are particles sized between 1 nanometer and 100 nanometers. We are investigating nanoparticles because they have special chemical interactions when they are irradiated by X-rays. These irradiated nanoparticles influence many reactions and the mechanisms behind these reactions. Specifically, my project focuses on gold nanoparticles and X-rays. Prior research has been conducted with gold nanoparticles and X-rays, and because of gold's physical and chemical properties, which allow for greater interaction with electrons. My project focuses specifically on the chemical properties as well as the chemical enhancement. X-rays are high energy and deeply penetrative compared to ultraviolet and visible light rays, which means that they produce a greater effect, and which means that they are able to produce greater effects. When pure water is irradiated by X-rays, it produces reactive oxygen species, or ROS. Solutions with gold nanoparticles are irradiated. The gold is able to facilitate the reactions involving ROS off its surface. My project's goal is to understand the gold concentration and ROS generation in order to maximize chemical enhancement. One reaction specifically that I will focus on is the hydroxylation of coumarin carboxylic acid, 3-CCA. 3-CCA, in the presence of OH radicals, a type of ROS, produces hydroxycoumarin carboxylic acid, or 7-OH-CCA. 7-OH-CCA is much more fluorescent, and this fluorescence, due to gold, can be quantified as chemical enhancement, as a greater concentration of gold would produce greater fluorescence. This is my fluorescence data, and on the left you can see that with greater concentrations of gold, the peak gets higher. On the graph to the right, which depicts enhancement by gold concentration, there's a consistent trend. At low concentrations, it is linear, but at high concentrations, saturation occurs and the enhancement is relatively small compared to at low concentrations. So fluorescence allows us to understand the beginning and the end of the reaction, but not anything about the mechanism of the reaction. One proposed mechanism in the literature is that the ROS generation is enhanced by gold nanoparticles, as seen in the red image. In the 1A step, there is no gold nanoparticles and relatively few ROS is produced. But in the 1B step, in the presence of gold nanoparticles, more ROS is produced. Our lab group's proposed mechanism is that the catalytic step occurs after ROS generation, and that the surface of the gold nanoparticles in the presence of oxygen facilitates the conversion of 3-CCA-OH adduct to the 7-OH-CCA product. Since fluorescence does not give us full insight of the mechanism, we must use electron paramagnetic resonance, or EPR. With EPR, we use a molecule called BMPO, and BMPO is a spin trap of hydroxyl radicals. This means that BMPO, in the presence of hydroxyl radicals, forms BMPO-OH adduct, which is able to be seen on EPR spectrum. There are many other spin traps, but we use BMPO because it is stable and has a relatively long half-life in comparison to the other spin traps. Finding enhancement through EPR. EPR is a form of spectroscopy, which allows us to quantify the amount of chemical enhancement. Specifically, I used the height of the second peak to quantify the amount of chemical enhancement. The red spectrum depicts the same blue spectrum, but slightly staggered to the right. Also, the noise is eliminated. When the noise is eliminated and each spectrum is staggered, it is easier to see and compare the second peak height of each spectrum. So for the EPR experiment, this occurred in 2 steps. One without DMSO and one with DMSO. DMSO is a scavenger of hydroxyl radicals, and without it, the ROS produced by irradiation is high, producing a high background signal. This background signal essentially masks the signal that we get from increasing the gold concentration, and we expected 0 to relatively few enhancement from it. But from the graph, the enhancement by gold concentration graph on the right, we actually see a linearly decreasing trend. This trend could be explained by scavenging of ligands, which increase in concentration with concentration of gold. The second part of the experiment is with DMSO. With DMSO, we are able to remove the background and analytically quantify the chemical enhancement. With the height of the second peaks, there's relatively no trend, but the data point still tends to 0. We hypothesized that with more experiments, all these data points would tend to 0. This is direct evidence of our proposed mechanism because if ROS generation occurred, there would be a linear increasing trend here, but we don't see a trend at all. And here we compare the fluorescence data with the EPR data, and specifically the without DMSO case, which is shown in red. Without DMSO, for the first step in the EPR enhancement, there's anti-enhancement. Yet overall, as seen in the fluorescence data, there is small enhancement. This means that the enhancement must be coming after the EPR probe, which implies that our proposed mechanism is correct. This research can be applied. There's still much more research to be applied in order to fully understand the mechanism. Once we are able to fully understand the mechanism, we will be able to design a material that would act as a catalyst to enhance yields of ROS, including hydroxyl-ROS. This catalyst would then be able to be applied to other reactions, including carbon dioxide reduction. X-ray and nanochemistry can also be applied to many other various fields, including nanomedicine, biochemical reactions, and energy efficiency. I would like to thank Dr. Ting Guo, Jennifer Lien, Rick, Egan, and Corinne, and the rest of YSP staff for their help and support with this research project. Thank you. Any questions?
Is there a difference between X-ray radiation as opposed to X-ray irradiation, and if so, what is that difference?
I think that they're the same because, yeah, they're the same. Logan?
How long did this reaction take?
So it takes a lot of time to prep the reaction, and for the EPR, for example, each scan takes about 20 minutes, and that's just for one sample. And if you can see in our EPR, there's many concentrations. There's even more. So we tested 0 ppm, 3 ppm, 6 ppm, 10 ppm, as well as 20 ppm in a different case. And we often ran duplicates, which means 2 of the same sample, and that took a lot of time.
How did you calculate the enhancement?
Okay. So we calculated enhancement by comparing each concentration of gold, whether for the EPR with the second peak height or the fluorescence with the main peak height. So we divided that value by the value produced by the 0 concentration gold, and then subtracted by 1. So which is shown in the enhancement by concentration.
I did EPR of organic molecules, so we use spin labels that attach to the sulfide group and cysteine. Did you have any equivalents?
So spin labels are the same thing as spin traps, I think. So we used BMPO, and there's also other various spin traps, but we use this one.
Thank you.
Next up, I would like to introduce Kevin Yang.
Kevin, we're running about 6 minutes ahead, so we're gonna just pause.
My people aren't here either.
Okay, so we're gonna pause. We still are, we're ahead by about 6 minutes.
Well,
We're ready anytime you're ready.
Hello everybody, my name is Kevin Yang, and I've been working in the Joe Laboratory under Kelly Chintabise, investigating differential gene expression between two distinct breeds of chicken, Fayumi and Leghorn, and their impact on NDV viral resistance. So NDV, or Newcastle disease virus, is a single-stranded RNA virus that infects the digestive and respiratory tracts of many wild avian species. NDV is transmitted through the exposure of healthy chicken to infected chicken. And since many chicken are raised in a confined housing area, infection runs rampant throughout the poultry population. Leghorn and Fayumi were chosen as our two breeds to investigate because past research on avian influenza virus between these two chickens revealed that when exposed to the virus, Leghorn had greater viral titer when compared to Fayumi, which supports the hypothesis that Leghorn are more susceptible to viruses when compared to Fayumi. This research also found that different gene pathways are being expressed. So this is extremely important because this suggests that the difference is due to a genetic factor and not just due to chance. So we believe that if we conduct this experiment on NDV, it will give us similar results, and this can give us a better understanding of the NDV virus as a whole. So 6 genes were identified based on their importance to the chicken immune response: TLR3, TLR7, IR7, INFAR1, CD8A, and PRKCD. So when the virus first enters your body, TLR7 or TLR3 will detect it. And TLR3 is responsible for detecting double-stranded RNA, while TLR7 is responsible for detecting single-stranded RNA. So after TLR7 or TLR3 has detected the virus, it will phosphorylate IRF7, which is the master interferon gene regulator, and this will stimulate the production of interferons. And these interferons will bind to IFNAR1, and IFNAR1 will stimulate interferon-stimulated genes, or ISGs, and these genes will inhibit viral replication. PRKCD is responsible for apoptosis in cells, and therefore is important for the inhibition of proliferation of the cells. And CD8A is an antigen found on killer T cells, and these killer T cells are responsible for removing infected cells from your body. So 100 chickens were prepared by subjugation to heat treatment and exposure to NDV virus. Tears, blood, and tissue samples were collected at 4 different time points: at no infection, 2 DPI, 6 DPI, and 10 DPI. So tears were used to measure viral titer, and blood was used to measure antibody levels. So this graph demonstrates that viral titer is much greater in Leghorn when compared to Fayumi. So this is similar to the results obtained in the previous study about avian influenza virus. This graph demonstrates that antibody levels were far higher in Fayumi when compared to Leghorn. So primers were developed for each gene of interest and were tested using a gradient PCR. Here are some of the components for PCR, and basically it's just a simple way of amplifying your target gene. qPCR is essentially the same thing as PCR, except it can measure the relative abundance of the targeted gene that you're looking for. So when you conduct qPCR, it will give you a CT value. And a CT value is basically the cycle at which the machine can detect a certain amount of DNA. So that number is recorded at this threshold cycle line across here. And each cycle, DNA is doubled. So a difference of 1 between CT values means that there's twice as much DNA present. And the CT value shares an inverse relationship with the cycle number, in that a lower CT value means that more DNA was present initially, because fewer cycles were required to amplify the DNA to a point where the machine can detect it, while a higher CT value means that less DNA was present initially because more cycles were required to amplify the DNA to the point where the machine can detect it. So these values were plotted on a 2 to the x graph to estimate the relative abundance of the gene being expressed. And 2 was used as the base because DNA is doubled every cycle. So here are the results of my qPCR trials. For these graphs, the horizontal axis are my 4 groups I tested: control leghorn, infected leghorn, control Fayumi, and infected Fayumi. The vertical axis is the relative abundance, the values of the CT values essentially. And the horizontal lines indicate that there is a statistical difference between 2 different groups based on a 2-sample, 2-tailed t-test with a p-value of 0.1. So for TLR3, for TLR3 in the spleen, there is a significant difference between the 2 infected groups and the 2 Fayumi groups. And this trend follows for TLR7 in the spleen and IRF7 in the spleen. So for CD8A, there is a significant difference between the control groups and the infected groups. For FNR1 in the spleen, there is a significant difference between the infected groups. And it's important to note that for these samples, specifically for the spleen, gene expression was higher in Fayumi when compared to leghorn, and after exposure to the virus, the gene expression level went up or was upregulated, while leghorn's gene expression level did not change or showed little change after exposure to infection. So for TLR3 in the lung, the 2 control groups, the 2 infected groups, and the 2 Fayumi groups showed a statistical difference. For PRKCD in the lung, the 2 control groups and the 2 infected groups showed a significant difference. For CD8A in the lung, the 2 leghorn groups and the 2 control groups showed a significant difference. And it's important to note how prior to infection, the gene expression levels for leghorn were far greater than after infection. So this suggests that NDV has the capability to suppress the interferon gene pathway. IFNAR1 for the lung shows statistical difference between the 2 control groups and the 2 infected groups. And here's a summary of all the qPCR trials I ran. The red squares indicate that there was a significant difference, while NS stands for no significance. So our results suggest that Fayumi have an increased resistance because the gene expression levels were far higher and they were upregulated, whereas in leghorn, they were not upregulated. So 2 possible theories were developed as to why this is occurring. And one of them is that, as mentioned before, NDV has the ability to suppress certain genes, specifically the interferon gene pathway. Similar studies were performed on other families or other viruses in the same family as NDV, and those viruses have the ability to suppress the interferon gene pathway. Another theory is that since Fayumi were originally from Egypt, they may have a higher tolerance to heat stress, and it's important to remember that chronic heat stress can impair your immune system, and our chickens were subjected to heat stress in this experiment. So, fortunately, further studies need to be conducted on polymorphism or replacement of nucleotides in the DNA sequence to determine how these specific genes are affecting the difference in resistance. What this research did do was provide candidate genes for this investigation of polymorphism. I would like to acknowledge Dr. Joe for allowing me to spend my summer in his lab, Kelly Chantabise for encouraging me even though I failed a lot of PCR runs, Dr. Pro Seilao and Dr. Ying Wang for their mentorship. She has 2 PhDs. Dr. Rick Pomeroy for providing such a great summer program, and Ann Hua, Stephen Ramsey, Corinne Rushing for giving me social advice. And Tyler McCurley for tying my ties for these presentations. And Jeremy for being Jeremy. Keep it up. I would also like to thank my roommate for allowing me to borrow his shower caddy every night, and the UC Davis Animal Science Department, and the Young Scholars Program here at UC Davis. Here are my citations, and are there any questions?
Well, what did you decide
So tears were extracted from the chickens by salting their eyes. We used an extraction kit, and the extraction kit gave us the values for viral titer. I know it's really bad, but
Why?
so we can measure viral titer in the chickens. Kate.
Why did you choose to use a p-value of 0.1 as opposed to 0.01?
Well, extensive research was conducted on these genes, and they all showed they all showed extreme importance in the immune system for chickens. So we thought it was justifiable to have more lenience when analyzing our data. Delaney.
If TLR3 detects double-stranded DNA, why would you use it to investigate NDV that's single-stranded?
Well, NDV is single-stranded, but at some point in the life cycle for viruses, they are double-stranded. So there may be some double-stranded NDV present in chickens, so we should look at TLR3. Renee.
Oh, why did you look at the lung and spleen as opposed to like other organs in the body?
Well, NDV is primarily a respiratory disease, so it would make sense to investigate the lung. And the spleen is, I believe, like the first lymphoid tissue. Yeah, well, the spleen is also affected from NDV, so those were the top two tissues to choose. Yeah. Michelle.
Can you explain how you labeled those graphs with the viral titer and the antibody?
Yeah. Right here. Okay, so the horizontal axis, or the vertical axis is basically just the concentration of virus. It's a really complicated unit, but that's basically what it is, just concentration. And the SP ratio is, so when you extract antibody levels from the blood, you measure them by fluorescing them. So the greater amount of fluorescence, the higher the value. So there's more antibodies present in beta. Nathan.
Question. How exactly did you extract the blood from the chickens from the chickens?
I believe, okay, I was not present for this part, but I believe they were just, well, I believe there's a kit.
To get blood out of a chicken, there's an artery just along your arm here, right? And so this is one of the main arteries for a human, just like in birds. So they have one that runs along your arm. So you just kind of find it, and the same way that you would do it with a human, you just prick it and blood comes out. Sometimes fascinating.
Yeah, what he said. I wasn't here when the chickens were. Willie.
What was the purpose of the heat stress during the procedure?
Oh, yes. So this research is like primarily important for Africa, because in Africa they lack the proper vaccines and treatments for these chickens. So in Africa, obviously it's really hot. So we wanted to simulate like what real life situation would be in Africa. Any questions? Thank you. I would like to welcome Renee Radushewitz to the stage.
Renee, is everyone who's coming here, we're about 2 minutes early.
Is Laura?
I'm not sure if she's coming.
And you know if Dr. Inouye is coming? He might come.
I'm not sure.
Okay.
Let's give him 2 minutes and then we'll.
You're welcome. Thanks for coming. Have a good drive home this
I know, man. Some people are rude about it.
past 6 weeks, I've been conducting my research in Dr. Inouye's lab under the supervision of graduate student Philip Day in the Plant Science Department of UC Davis. And my research was to compare the targeting mechanisms of TOX75 and OEP80, which are two proteins within the outer membrane of the chloroplast. But before I start talking about my research, let me discuss the skill of pipetting. Pipetting is a commonly used skill within biology and chemistry research, but most people tend to forget that this is an imperfect tool, in that it can vary in reproducibility and in accuracy. Reproducibility corresponds to how well the pipetter, such as myself, can match the ideal amount that we're trying to measure, while accuracy corresponds to how well the pipette itself can match the ideal amount. And we must keep this in mind because when we're taking measurements, what we're hoping to measure may not actually be what's happening in reality. And before I start talking about the proteins within a chloroplast, let me begin by starting off about the origin of chloroplasts themselves. So chloroplasts originated from the endosymbiotic relationship between a cyanobacterium and a eukaryotic cell. So the cyanobacterium was engulfed by a eukaryotic cell, and this resulted in a gene transfer. So the genes from within the eukaryotic cell were transferred to the nucleus of the eukaryote. But this presented a problem to the cyanobacterial ancestor of the chloroplast because by having the genes inside the nucleus, this meant that the proteins that these genes encoded would be produced in the cytosol of the eukaryotic cell rather than within the chloroplast ancestor. So the result of this was OMP85, or outer membrane protein 85. Outer membrane protein 85 and its homologs have been shown to be associated with assembling proteins within the outer membrane of the cyanobacterial ancestor. And homologs of OMP85 are commonly found within gram-negative bacteria, mitochondria, and chloroplast as shown here. So in here, it would be within the outer membrane in this chloroplast. Homologs of OMP85 are distinguished by their N-terminus, potra, and beta barrels. In my specific research, I'll be looking at the homologs OEP80 and TOX75, which are descendants of OMP85. So TOX75 has evolved to carry the function of transporting proteins within the chloroplast's outer membrane. On the other hand, the function of OEP80 has not been demonstrated experimentally, but is hypothesized to match that of OMP85. Now the N-terminus of TOX75 has been found to be associated with targeting the chloroplastic envelope, and it's assumed that the N-terminus of OEP80 does the same thing. So to begin my research, I will create a chimeric gene involving the N-terminus of TOX75 and replacing that of the N-terminus of OEP80. And this will allow me to compare the difference in targeting mechanisms between TOX75 and OEP80 since the N-terminus is associated with targeting. And at the end of my construct, I will include a T7 tag, which will later be used to detect the proteins being made from this chimeric gene. So before I'm able to create my construct, I start off with two different templates, TOX75 and OEP80. These red arrows here and here indicate primers that will be used to amplify the portions between the red arrows to create this entire construct above. So this portion will match here, and this portion will match up here. So before we actually get to the full construct, we have to amplify these specific portions of TOX75 and OEP80 through PCR. And we'll take the N-terminus of TOX75, and we'll take the potra, beta barrel, and T7 tag from OEP80. And this is what the entire construct should look like after a second PCR is conducted. So in our initial attempt at PCR, we expected a band of about 2,500 base pairs, but the first time that we tried that, if you look closely, you can see a really faint band. And this faint band indicates that there's a low concentration or low amount of the construct that we produced, which is not ideal in my research. So we decided to look for a better way to create our construct by finding the ideal PCR conditions. So the conditions that we tried were variations of PCR annealing temperatures, 52 degrees Celsius and 62 degrees Celsius. And also the other conditions were the difference in templates, which were purified portions of the DNA and unpurified. Unpurified portions would mean creating the construct like how we did the original attempt, while purified would be creating that construct and then cutting out only the sections of DNA that we need. But if you look here, the unpurified portion at the 52 degree annealing temperature produced the clearest band, indicating that it had the highest amount of construct and is the correct construct at that. So that's the conditions that we followed. However, when we tried to follow these ideal conditions, the first time we did not see any band, which should be right about here. And this is likely because the substances were not added correctly. And as I mentioned earlier about pipetting, there could have been an issue with the accuracy and reproducibility of this amount, which could have led to have no band in this corner. Because when we reproduced our conditions again, we were able to get this band, which is likely because the correct amounts of substances were added to create the construct. So once we had this chimeric construct, we were able to perform a BP reaction. So what a BP reaction does is it takes our construct that we created and inserts it into a donor vector, which is a different plasmid. And this is the donor vector that we used here. And my construct would be replacing the portion from here to here. So once that newly made plasmid is created, we'll transform it into E. coli, which will then be plated. To ensure that the colonies that grew on the plates did in fact have the construct that we created, we performed colony PCR with 14 different colonies, and then the last 2 tests were negative controls. Now these negative controls were not supposed to show a band, but they still did, which is likely because of contamination or by the slight chance that a different band was produced, and it's not the desired construct. So for negative controls, my first negative control right here was supposed to be a different insert, which should not have been amplified through PCR because the primers would not have matched. And then my last negative control here was a blank, where I did not include a colony at all, so nothing should have been amplified. So since our negative controls came up positive, we had to conduct a second colony PCR with the same 14 different colonies, and then the last 2 were the same types of negative controls. And while one negative control did produce what we expected, which was no band at all, the last negative control still produced a band, indicating some sign of likely contamination. So we ran a third set of colony PCR with the same 14 different colonies, and the last 2 were the negative controls. And as you can see, most of our colonies did have the band, but for example, like lane number 4 does not have a band indicated, which means that there happened to be a plasmid growing that did not have our construct. And for our negative controls, you can see faint bands, which are much better results compared to what we had earlier, compared to the very intense bands that showed that there was some construct present. But these faint bands are much better and more likely because of contamination or spilling over from the wells from gel electrophoresis. But since we had these faint bands appear where there should have been no band, we had to double check that the plasmids are created correctly, once again through restriction enzyme digestion. So what restriction enzyme digestion does is it utilizes the restriction enzyme known as EcoR5, and it creates cuts in the plasmid at the cut sites. So these lanes with the pluses are places where we added the restriction enzyme, so we expected multiple fragments. In particular, we expected 2 fragments. And the signs with the negative sign above them mean that we did not add the restriction enzyme, so we expected just a single band because no cuts were made. So the first time that we ran it, we only had single fragments, which was not what we were expecting. We were hoping for multiple fragments in the lanes with a plus sign. So this is likely because the enzyme EcoR5 was not added, and this traces back to the fact like the pipettes are not a perfect tool, especially since the amount of EcoR5 is very small. I believe it's 0.5 microliters. So it's a very small amount, so if you don't get that amount in there, you won't see the expected bands. But once we tried it again, we were able to get the correct fragments, as indicated here, and we were able to move on with this process. So once we verified these plasmids through that colony PCR, we were able to carry on with an LR reaction. And this goes back to like the BP reaction, where we produced an entry clone. And that entry clone had our gene within a different plasmid. But this time in the LR reaction, our gene will be switching with a different gene within something called the destination vector. So we will be looking for an expression clone, which contains our gene and then the portion of the destination vector here. And in that destination vector portion, there will be a sequence encoding resistance to an antibiotic. So when we transform this plasmid into E. coli and plate them onto a plate with an antibiotic, we should only see expression clones because they have the resistance to the antibiotic on the plate. Thus, when we plate them and have E. coli with these plasmids, we should only find colonies that contain the plasmid with our gene and the rest of the destination vector. And again, we had to verify this through another type of colony PCR. This time we only used 4 different colonies, and then these 2 were negative controls. One had a different negative. This negative had a different destination vector, which also indicated that nothing should have been amplified, and that's why it should be blank like it is. And this lane here was also expected no band because we did not put any colony at all, so nothing was there to be amplified. And this last column right here was a positive control, which is supposed to produce a similar sized band as what we expected from the first 4 colonies that we tested. And since the negative controls did not produce any band, and the positive control matches along with our colonies, this indicates that we did, in fact, produce the correct plasmid with our construct within it. So that concludes my lab work for this project, but to continue on with this specific project, we would isolate these plasmids and then transform them into agrobacteria. And then once we do that and allow the agrobacteria to grow, we can allow them to infiltrate the leaves of Nicotiana bethamiana and then isolate the chloroplast within these leaves. Then we can perform SDS page and a Western blot to analyze the proteins produced by the gene that we created. Further along the line, after all this has been, all the previous steps have been completed, we could perform similar types of experiments using different constructs involving different combinations of TOX75 and OEP80 with the T7 tag. So these genes could be expressed, and then we can compare their targeting mechanisms to have a better understanding about TOX75 and OEP80. So I would like to acknowledge Philip Day, my mentor, for guiding me throughout the process. I'd like to thank Dr. Inoue for enabling me to be a part of his lab, Jeremy for being my counselor and supporting me along the way, the YSP program for letting me be here in the first place, and all of you guys for being such great, intelligent people. And special shout out to Jackie Ho, my roommate, for moral support. And lastly, I'd like to thank my parents in the back for enabling me to be here and for their constant encouragement. These are my references. Are there any questions? Any questions?
What's the difference between classical PCR and colony PCR?
So classical PCR is like, when you're creating the initial construct, it only amplifies like the portion. They're both essentially like the same process, but in colony PCR, we're using bacterial colonies, essentially. Any other questions?
Once you get to inserting the bacteria into the plants, what are the proteins supposed to do?
Oh, so the proteins will be expressed, and I believe they should. We're hoping that they would target the outer envelope of the chloroplast, I believe, and then it would ensure that they were targeted there by running SDS page and Western blot to see if they were targeted the chloroplast correctly.
Okay, thank you.
Please give a warm welcome to my colleague, John Alzon.
Good afternoon. My name is John Almazan. I have been performing research in Dr. Drakakaki's Plant Science Laboratory under the mentorship of Dr. Michelle Ruiz-Rasquet. Primarily, my research involves experimenting with Arabidopsis mutants that are hypersensitive to ES7. But before I describe my experiment, let me first describe some of the processes that are directly involved in my research. The first term is plant cytokinesis. Plant cytokinesis occurs during plant cell replication, in which the cytoplasm of a new dividing cell splits in between the chromosomes of two daughter cells. Plant cytokinesis, which is different from animal cytokinesis, involves the formation of a new cell plate, which later matures into a new cell wall. So the formation and maturation of the new cell plate involves vesicular trafficking, where vesicles deposit polysaccharides and other molecules to the cell plate. Some of these polysaccharides that are deposited at the cell plate include callose and cellulose. Callose is a polysaccharide that is thought to provide mechanical support and stabilization to the membrane network at the cell plate. Also, based on past research from Dr. Drakaki's laboratory group, it is hypothesized that callose is deposited in the later stages of cytokinesis. In the diagram to your left, we are comparing the initial stages and the later stages of cell plate maturation. In this diagram, callose is represented by the orange circles. As you can see, the initial stages do not have any callose deposited at the cell plate, but there is callose deposited in the later stages of cell plate maturation. So one way to see callose depositing at the cell plate is by confocal microscopy. The image to your right here. The image to your right, we can see these dark blue stains, which is callose depositing at the cell plate during that time the image was taken. So the second term I would like to explain is ES7. And ES7 is a small molecular chemical that specifically inhibits the callose deposition process during cell plate maturation. And these three pictures here, we are comparing different concentrations of ES7 and their effect on the callose deposition. Now, callose deposition is represented by the pink lines here. And as you may see, that as the concentration of ES7 increases, callose deposition becomes affected. As we see, there is a lot of gaps in between these callose depositions. So since ES7 inhibits callose deposition at the cell plate level, we can see the overall root growth reduction in the overall root. There's a trend that in increasing amounts of ES7 concentration, there is a reduction in its growth. Further, Dr. Drakaki's laboratory uses ES7 as a tool to identify specific proteins that assist callose deposition at the cell plate. In Dr. Drakaki's lab, there are many mutants that are needed to be confirmed to see if they are resistant or hypersensitive to this chemical ES7. So the primary goal of my project was to confirm previous studies in which that identified Arabidopsis mutants that were hypersensitive to ES7. Specifically, I was researching 3 mutants, which are called 15-1, 32-2, and 81-2. And all 3 of these mutants are part of the Arabidopsis species and are included in one ecotype called the Columbia ecotype. So in order to confirm the hypersensitivity of these mutants to ES7, each mutant and some parental seeds, which are called RAB2A, were grown in 2 sets of plates, one of the plates containing DMSO and the other group of plates containing 2.5 micromolars of ES7. And so both types of seedlings were incubated and grown in an incubator at 22 degrees for a maximum of 4 days. And at each day, I would scan the plates with an Epson scanner, and later I would measure the individual seedlings using a computer software, ImageJ. And after I obtained all of the measurements for the individual seedlings per day, I used an online statistical analysis called GraphPad to detect significant differences between the root lengths in ES7 and the root lengths in DMSO. So based on these findings with the statistical analysis, it showed that there were significant differences between seedlings in DMSO and seedlings in ES7 starting in day 2 after germination. There was
day 2, day 3, and day 4 showed extremely significant results. So for the main results, I created a graph with relative values of the root length reduction. And in this graph, the value 1 represents the parental line control. So since we're measuring the reductions, values that are above 1 means that it is hypersensitive to ES7. Values that are below 1 means that it is resistant to ES7. So based on this data, it is proven that the mutant 81-2 is hypersensitive to ES7. The other mutants, 32-2 and 15-1, proved to be resistant, surprisingly, to ES7. And as we can see in the hypersensitive mutant 81-2, that the root growth length was greatly reduced because of this ES7 chemical compared to its control group in DMSO. Okay, so after learning that 81-2 was a mutant hypersensitive to ES7, we wanted to go to the confocal microscope to see the cell plate formation and the root morphology to see if it was disrupted by the ES7 chemical. So what we were expecting to find before looking in the microscope was this. We expected that the DMSO treated seedlings would exhibit normal and perfectly fine root morphology, and that the ES7 2.5 micromolar would exhibit abnormal and would be defective because of the ES7 chemical. But instead, we found that there were not really any significant differences in the root morphology itself. The ES7, which was predicted to have abnormal root morphology, actually exhibited pretty normal morphology compared to its control group. Also, we looked at cell plate maturation under the confocal microscope, and this is what we had expected to find. We had expected that in the seedlings in DMSO, there would be normal and healthy cell plate maturation. And we had expected that the ES7 2.5 micromolar would exhibit gaps and defected cell plate maturations. But instead, we did not find any significant differences in the cell plate in the ES7 2.5 micromolar and the DMSO compared to the RABI2A parental line. So based on these results, it is suggested that smaller concentrations of ES7 could affect the root growth, but stronger concentrations of ES7 are needed to impact and alter the cell plate formations and the root morphology. Currently, the knowledge of the molecular level of cytokinesis is very limited, and so experimenting with ES7 and Arabidopsis mutants provides greater knowledge about the callus deposition process and the later stages of cytokinesis. And in the future, we will look at the specific mutants that tested to be hypersensitive to ES7, and we will look at those mutant genes that are altered by the mutations, which will eventually lead to discovering new proteins and other influential factors that contribute to callus deposition and contribute to the later stages of cytokinesis. I would like to thank Dr. Georgia Drakakaki for allowing me to perform research in her laboratory and for guiding me and always welcoming me to the lab. I would like to thank my mentor, Dr. Michelle Ruiz Rasquet, for always welcoming me to the lab and teaching me new laboratory techniques. I would like to thank Dr. Rick Pomeroy and the Young Scholars Program for providing us with this wonderful opportunity to do research at UC Davis. And lastly, I would like to thank my counselor, Stephen Ramsey, for always there for supporting us and for answering any of my questions about the research. These are some of my references. Are there any questions about the topic?
Is there a reason why you looked at root growth instead of like stem growth or anything like that?
Right. Well, the root growth is like the main part of the plant that grows. So we're looking at the tips of the roots for root morphology and so.
What's the significance of root morphology?
Right. Well, it shows if ES7 had an effect on the roots. if ES7 did have an effect, that it would show abnormal morphology.
In your graph, is there a reason you didn't average the 3 bars on value?
This one? Okay, yeah. Yeah, well, one of the things that we had to do was that ES7 is very variable. And from replicate to replicate, each one of these bars represents one plate or one replicate. And each one of those had a pretty significant difference. So to put it in one average, that would have been not good for the results. It would have affected the overall results. Any other questions? Thank you for listening to my... Thank you for listening to my presentation. Now I will introduce Alex Shu.
Alex, we're running about 2.5 minutes ahead. Is everybody who?
Yeah.
Okay, and then do you have anyone watching at home?
No.
Okay, then when you're ready.
Hello everyone, I'm Alexander Shu, and over the past 6 weeks I've been doing research in the Biomes Lab under graduate student Mo Coakley and Dr. R. Holland Cheng, both of whom are part of the UC Davis Department of Molecular and Cellular Biology. I've been studying more efficient and automated ways to create accurate virus models. So our lab has generally decided to study viruses and other similar macromolecules because a greater understanding of these concepts could lead to significant medical breakthroughs. For example, understanding the infectivity of viruses and how exactly the structure works in relation to how they can infect could allow us to better combat them. On the other hand, we can use these infectious properties of viruses in our own virus-like particles so that they may act as nanocarriers and carry our own engineered medicine directly to diseases in the bloodstream. We've made a lot of significant developments when it comes to reconstructing viruses. For example, we know about common lines and the icosahedral lattice of many virus particles. These give us a good idea of the orientations and the structure that many viruses will follow. And this knowledge, combined with computer algorithms that have been developed over the years, have allowed us to create these 3D models and reconstructions. We've also learned how to create class averages, which are essentially groups of similarly oriented particles that give an example of the orientations one might find when you create an actual 3D reconstruction of the virus. So, in the steps of creating class averages or reconstruction, there are 3 general steps. The first step is selecting the particles from each individual micrograph, generally taken with a transmission electron micrograph. This step is known as boxing. The second step is to analyze the orientations of the particles selected. And then finally, we take the particles and render them into either the 3D image or the class average that we're looking for. So the problem that we're encountering right now is with manual boxing, the very first step. Single-handedly going through hundreds of micrographs to look for thousands of particles is simply very slow and tedious, and so if we can find a faster way to do this process, it will be much easier for us. During the project, I tested 2 different methods of semi-automated boxing, and I compared them to how they fare against manual boxing. During automated boxing, you're also presented with a sort of sliders that you can adjust for each individual micrograph. We also had wanted to initially try to optimize these sliders. However, we quickly discovered that since each micrograph varies too much from micrograph to micrograph, this would not be possible as there would be no set of best values that would work for every individual micrograph. This is just an example of what boxing looks like. On the left, we have our micrograph and boxed particles, and on the right, you can see the particles up close. These were particles of hepatitis E virus-like particles, or HEVLP for short. And these particles were boxed at a box size of 256 pixels and a resolution of about 1.7 angströms per pixel. Now, the 2 automated methods that we tested in comparison to our manual control are known as autoboxing and autoboxing from references. The difference between these 2 methods is that the former requires you to select individual particles in the micrograph And then from there, it will compare all other automatically selected particles to the particles in the micrograph that have already been selected. And the other one, autoboxing from references, allows you to pull references from an outside source so that it compares particles that it wants to select from the outside instead. So this is just an example of hepatitis E virus. These are refined class averages that we might want to find after many refinements. These were created from a 3D reprojection of hepatitis E that was then turned into 2D images, which is why we have a lot of clarity, and you can see the protrusion, middle, and shell domains of the class average, which I'll be getting into later. So for a manual trial, you'll notice that we use the selection set of around 400 particles, and the class averages themselves are indeed quite blurry. This is to be expected because we need around 1,000 or more particles to have very clear class averages and a good reconstruction. However, you can still notice a lot of the details on the class averages themselves, and for the most part, they're pretty high quality. On the right, I just have a comparison of class averages of about 1,200 particles and what you might expect if you did this experiment with a lot more particles. So when we were testing the other automated methods, we first ran each of the 2 automated methods without going through and deleting any of the bad particles, just to see if the automated method could do well without human interference. Afterwards, we ran both of them again. However, this time we simply deleted the particles that were not suitable for either class creation or particle reconstruction. These are the results when we use the first method, autoboxing. The first thing you might notice is that in one of the autoboxing that wasn't optimized, one of the class averages, it is completely fuzzy. And this is to be expected because we didn't go through and optimize it at all. Therefore, we must have picked up a lot of outliers and particles that weren't necessarily particles along the way. However, for the most part, it still is about the same resolution as the class averages created when doing manual boxing. And optimizing the process does seem to have improved the average value, the average appearance of the particles. So qualitatively, this method, whether human interaction was involved or not, this method is a success. So for autoboxing from references, these were the references we decided to use. These were also hepatitis E virus classes created when taking a 3D reconstruction of the virus and then turning it into 2D class images. However, you'll notice that these are just inverted, which helps with the particle selection process. The class averages we created when we did this method are about the same as the previous 2. You'll notice that there is no very glaring obvious inaccuracies during this time from both the autoboxing references and the optimized version. But for the most part, all of this suggests that these viruses, whether they're automatically selected or manually selected, they produce about the same results. And that they're relatively clear. They still have that fuzziness because there are only 400 particles in the particle set. However, it appears to have done relatively well, and this can be significant because that way we can do automation rather than relying on simple manual labor. This is just the raw numbers of going through the particle selection process. I tried to keep the data set for each method of boxing around 400 particles so that class averages and reconstructions later on could be accurately compared. I also tracked the number of particles that were removed during the optimization periods of both different automated boxing. And you'll notice that autoboxing from references appears to have picked up slightly less for particles compared to those picked up by just normal autoboxing. These are some of the criteria we used when we were looking for bad particles that we want to remove during the optimization process. In green, we have the proper particles that have been properly boxed. You'll notice that the faint red square around it is perfectly centered on it, and there's no debris and there's no other particles around it. Three of the main problems we encountered when we were doing automated boxing are highlighted in blue. In the top left, we have particles that simply weren't picked up because the automated software just couldn't recognize them. In the top right, we have particles that just aren't virus particles at all, and so we're going to encounter some debris, like in the autoboxing class average perhaps. And in the bottom left, we have a properly boxed particle. However, since its proximity is too close to other particles in the micrograph, this would not be suitable for 3D reconstruction or class average creation because there would be too much background noise. This is an example of a 3D reconstruction of a virus. In red, you can see the intrusion domains. In yellow are the middle domains, and green and blue are the shell domains. Notice how the structure of the virus is pretty clear and sort of looks similar to the class average we've seen, except this is in three-dimensional form. Since hepatitis C virus is an icosahedral virus, we have five-fold, threefold, and two-fold symmetry, and these are represented by the pentagon, triangle, and line. So you can clearly see some of the traits that make this reconstruction much easier. Our manual reconstruction appears to be about the same quality as that of our example reconstruction. It might be a bit fuzzier, it might be a bit rougher, but for the most part, most of the details are maintained. And while the orientation might be flipped a bit, that can just be solved by just rotating the image a bit more. Autoboxing and optimized autoboxing form equally good 3D reconstructions. They still share the same orientation flip, but for the most part, all three domains of the virus are there, and so this shows that 3D reconstruction does have some success with automated methods. However, when we get to the autoboxing from references and optimized version of that, we notice very large discrepancies comparing this to previous other models created. For example, if you look in the autoboxing from references model, you'll notice that some of these particle growths are floating in midair, which is not physically possible. And so we do have some similar structures, like this bridge-like structure that goes over some space. However, these are just not very similar to the 3D reconstructions that would be there. Obviously, with further refinement, these models will turn into those perfected models that we would like to see. However, at this initial stage, we have realized that the small amount of refinement does not exactly work for all forms of 3D reconstruction. However, we can still tell that automated reconstruction does seem to be able to produce reasonable enough 3D reconstructions for any other experiments that may be done on viruses. One main important point we have to consider is that these reconstructions and class averages were collected with only around 400 particles. As I've mentioned, we need around 1,000 or more particles to create an accurate class average or an accurate 3D reconstruction. So that should be taken into account when we are looking at if this is truly a success. We can also take into account the references we used, which were perfected classes, and they differ a lot from the grainy images that we saw in the micrographs. So that may play a part in resolving the three main boxing issues. There are also many different programs that we can use for boxing and 3D reconstruction. Maybe that can also play a role in solving these errors. I'd like to thank Dr. Chang and especially Mo, who's back there, for making this all possible, as well as the rest of the science lab. I'd like to thank Stephen, Rick, Megan, and the rest of YSP for being here for me always. And I'd especially like to thank my parents for making this entire thing possible. Thank you.
How does optimization technically work?
Optimization it's just manually going through the particles that were automatically selected and deleting the ones that do not fit the criteria of the good particles. Henry?
Why did you guys choose hepatitis C of all viruses?
We ended up choosing hepatitis B virus as opposed to other viruses because in addition to it being icosahedral shape, which makes it easier for reconstruction, we also have worked with it in the past, and so we have a general knowledge of how 3D reconstruction could turn out. Really?
Why do you think there's such a big discrepancy between the 2D pictures of the automated with references versus like the 3D model that is completely off?
I'm not entirely sure. My best theory on it is probably because during the class average creation, since you're averaging a bunch of particles, you don't necessarily see any of the bad particles. It's just like a general form of what the orientation should look like. However, there could have been a lot more particles that weren't necessarily good for reconstruction that could have snuck in and ended up ruining the reconstruction. Back?
How many particles did you need to delete or modify during the optimization step, generally?
In general, we deleted around 40%.
40%? Yeah.
We deleted about 40% for both of them. It appears that autoboxing from references appears to pick up less bad particles, but for the most part, it's pretty similar.
When analyzing the different different viral structures, there was definitely like basis you guys followed, but how did you compensate for the esoteric qualities with each virus?
I don't follow. Can you rephrase the question?
Okay. Can you go to the slide of one of the virus? So there was like, so you guys follow like a certain model on kind of how you guys should analyze the data you were receiving, but was there any way like you guys compensated for like the specific like qualities within each virus viral model?
We mainly have to rely on like what the particle looks like, and then most of the work is done by computer programs like EMAN or electron micrograph analysis, and so they take care of most of the like analysis and like orienting. So I'm not entirely sure what goes on too much about the background technical aspects of that, but for the most part, it's all done for us.
So is auto-boxing or like the optimized version of it become more accurate or less accurate for different like strains of viruses?
It could potentially, because we are currently using and looking at icosahedral-shaped 3D viruses, which are very predictable. Their structure follows that simple geometric fashion. And so if we ended up using like more viruses that are shaped like a strand or viruses that aren't symmetric, then we could encounter more errors. Any other questions?